Hello, my name is Amari Smith, and today I'll be talking about the Salinization of Soils Lab. As always, the first step is always to make sure you got your safety goggles and your gloves. So I made sure that I had mine on. I actually ripped a hole through the glove as I was putting it on, but luckily I had extra pairs in my closet, so I was able to keep it chugging. All right. The second, well, really the first step in the actual lab is was to label the lids of the Petri dishes: 0.0%, 0.5%, 1%, 1.5%, and 2% with the permanent marker. Um, Next was use scissors to cut paper towels into 10 discs of the same diameter as the Petri dish bottom. This was particularly kind of challenging for me. I don't know why, but I've always had a bad relationship with scissors, and this was no different. I I don't know. It took me a while to uh, um, cut it just the way it was supposed to, but I got through it. The next step was place 2 paper towel discs in the bottom of each Petri dish. Then evenly distribute 10 beans on top of the paper towel disc in each Petri dish. And I will have pictures of this process included in my PowerPoint presentation. Um, Next, prepare the 2.0% saline solution by placing the 100 milliliter glass beaker on the scale and tear the scale. Then weigh 1.0 gram of salt in a 100 milliliter glass beaker. And then remove the glass beaker containing the salt from the scale. Add distilled water to the 25 milliliter graduated cylinder until it reaches 20.0 milliliter mark. Use a long, thin pipette to add distilled water to the graduated cylinder until it reaches 25 milliliter mark. I did. I went through this entire process, and this from this process was relatively easy. That was probably the easiest part of the lab for me. I didn't really have too many issues in that regard. Let's skip down to the questions from the lab. The first question was explain why salinization is a concern for agriculture around the globe. My answer to this was excess salts reduce plant growth as well as limit their ability to uptake water. The second question was what was the purpose of using distilled water instead of tap water in this experiment? And I said that distilled water is free of contaminants and minerals that could affect the growth of the bean. The third question was why were the Petri dishes containing the beans weighed at 24-hour intervals? And I said that the seed has to be completely hydrated, so the 24-hour interval allows for the seed to uptake and retain that water. The next question was, what was the minimum salt concentration that affected bean germination in your results? And I said 1%, as seen in data table 1, which I will be providing them the presentation to give you more of a visual perspective. But yes, I said 1%, as seen in data table 1. There was no germination of the beans in that dish. The last question was, based on your results, would you categorize the bean species used in the experiment as a halophyte or a glycophyte? Include the definition of each term in your answer. And I said glycophyte because they did not produce in the solutions with more NaCl. Now I'm going to kind of talk about the data that I was able to collect from this lab. So for the 0.0 cup with the beans, I got 30.71 grams. For the 0.5% cup, it was 31.05 grams. For the 1.0% cup, it was 29.92 grams. And for the 15% cup, it was 30.00 grams. And for the 20% cup, I got 30.91 grams. The chart that I will be providing in the PowerPoint presentation will give you more of just a better understanding, or just a better visual of how it changed over the span of 5 days. And that's pretty much the gist of what I have to talk about. Everything else will be included in the PowerPoint, as I said. But this lab was very fun for me. Once again, challenging. But I learned a lot at the end of the day, and I think that's what it's really all about. I think when I look back on it, I think I'll probably appreciate this type of lab over the others, because it wasn't easy, you know what I mean? And it required me to kind of get outside of a box a little bit, and just be creative a little bit, and force myself to learn more. And thank you.